Uh, Hello everyone. In the previous video we discussed about the light reaction and how and what were the products which we were able to gain as the result of that. And just for a review, in the light reaction we actually so let me write it first down. In the light reaction in the light reaction we actually, let me make it better. In the light reaction we essentially used sunlight. We used sunlight and as a result of sunlight we were able to generate we were able to make water, and along with water we made some ATP molecules and some NADPH. Now we made all this and essentially we used sunlight, and there was also water here, so these hydrogens got detached from this water, and we were able to reduce this NAD plus into an NADH, and eventually we end up having, so then this oxygen left on it combined with another oxygen to make us and to give us an O2, and you can see here that we still don't have our glucose, which the which the photosynthesis equation tells us. So you have carbon dioxide, you have water, and you end up with glucose. H12O6. You end up with glucose and oxygen. So we have the oxygen here, but and we use the water, so you can eventually say, okay, now we're going to use the carbon dioxide to end up having a glucose molecule. And you're absolutely right. And just for balancing it out, so we would have 6 of this, 6 of this, and 6 of that, right? 6, 6, yep, that's right. Now in the dark reaction, or the light independent reaction, remember that it's called dark reaction because it necessarily doesn't need light to occur, but it does occur in the presence of light. So it would be better if we would just call it the light independent reaction, or the Calvin cycle, named after the scientist Kelvin, obviously. The Kelvin cycle. Now what happens is, actually in this process we take the products from the light reaction, and the products which we take are ATP and the NADPHs, and these two essentially give the energy, or are the source by which the Kelvin process occurs. And what happens is that, just to give you an overview of what happens, is that you essentially have carbon dioxide and an ATP molecules and NADPHs. Oops, that doesn't look like an N. Yeah, there it is. So you have ATP molecules and NADPHs. So these three essentially go into the Kelvin cycle, and you end up with a G3, that's a 3G3P molecule. And remember, if when we discussed about glycolysis, we eventually end up with having a G3P, which is glyceraldehyde 3-phosphate, when we split glucose into two parts. because glucose is a 6 carbon chain molecule. So 1, 2, 3, 4, 5, 6, and if it has a phosphate group attached to it, and remember at that time it was fructose 1,6 biphosphate, and when we broke this thing in half, so we had two, so with these things are now in half, so we have two 3 carbon chain molecules right here, and each of these were called G3Ps, which had the which had the energy, which had enough energy stored in them, and which were able to, and from these things we were able to make pyruvates, and eventually we end up having 2 ATP molecules. So that was all in glycolysis, which we studied. But in this part we end up at G3P molecule, and when we have two G3P molecules, so you can imagine as going in a reverse process, these two G3 molecules join together, and we essentially end up having a glucose molecule which goes into the mitochondria, and through cellular respiration we gain ATP. Now, so this is an overview. Carbon dioxide, ATP, NADPHs, they all combine together and enter the Calvin cycle, and we end up having a G3P molecule. Now, so like in the previous video when we studied the Krebs cycle, which was a part of cellular respiration, and we discussed why is it called the Krebs cycle, and the first reason was, okay, it's called a cycle because we regenerate, we get the things which we put in. We eventually end up having the same thing. So because we start off with a molecule, it goes through a series of processes, and in the end we get that molecule back. So that's why we call that the Krebs cycle a cycle. So now here we call it the Calvin cycle. So why do we call it the Calvin cycle? Because of the same reason. We start off with a molecule, and it's written like this: R U B, that's a B, RUBP, and what this molecule is, it stands for ribulose biphosphate, which is a 5 carbon chain molecule. And what happens is that this carbon chain molecule right here attaches with carbon dioxide, and enters into the Krebs cycle. And when it's a lot of processes going on here. So if I were to just write it more better, this isn't, the carbons are all mixed up. So the ribulose biphosphate molecule, or it's sometimes called ribulose 1,5 biphosphate, and I'll tell you in a second why it's called that. So you have 1, 2, 3, 4, 5. So these are the 5 carbon chain molecule, and you have a phosphate group attached to number 1 carbon and on the number 5 carbon. So that's why you call it the ribulose 1,5 biphosphate, or ribulose biphosphate. And it joins carbon dioxide, and you eventually end up having a PGA molecule, which is called phosphoglyceric acid. Now, what happens is that so if you start off with so these molecules and join together to form a PGA molecule. This PGA molecule goes through a series of processes, and it eventually ends up being called what we call a G3P molecule. A G3P molecule, which is the glyceraldehyde 3-phosphate, and this glyceraldehyde 3-phosphate, some of it is it's used, and some of it is gone back to becoming again a ribulose biphosphate molecule. So you might be thinking, okay, how this process occurs. What happens is that at the first stage, when you start off with ribulose, when you start off with RUBP, or the 5 carbon chain molecule, so this is the 5 carbon chain molecule right here, and you have carbon dioxide with it. So these two molecules join together with the help of enzymes. So let's say we have three molecules of RuBP and three molecules of carbon dioxide, and these two molecules are react with each other with the help of an enzyme called Rubisco. So as these things join together, so you are able to generate about 6 molecules of PGA or PGA, which is a 3 carbon molecule with a phosphate group attached to it. So 6 times 3 is 18, 5 3 is 15. Yep. So we have 6 molecules of PGA. Now this PGA molecule, with the help of, you can imagine its structure is quite similar to that of the G3P, but it's the carbon is same, but it has different, that the structure of hydrogen and oxygens are different. So that's why we named them differently. And so what happens next is that with the help of ATP and NADPHs, which we were able to generate during the light reaction, so we use 6 ATP molecules. Well, not we, the plant uses 6 ATP molecules and end up with an ADP molecule plus a phosphate group. Along with that, we are making the NADPH is reduced to NADP plus. That's a plus. Now you might be thinking, okay, why am I giving energy to this molecule and why am I giving energy to this molecule? Because if you know why this process is occurring, in order that we make glucose, and glucose is a highly reduced molecule because it has a lot of hydrogens in it. Basically it has 12 hydrogens and it has a lot of hydrogen in it, and its electrons store a lot of potential energy. So when its glucose is used in the cellular respiration, it also liberates a lot of amount of energy. So that's why we are just this main function of this process to keep reducing the molecule, that then we make glucose, so we have a highly energy rich molecule at our hands, that which we can use to generate ATP as a source of energy. So you use 6 molecules of ATP and eventually end up with an ADP and a phosphate group, and 6 molecules of NADPHs, and they are reduced to NADP plus. So eventually end up with having a 6 molecule of G3P. Now, out of 6 of these molecules, 5 of these molecules do not leave the cycle. So 5 G3Ps are still in the cycle. Only 1 molecule leaves the cycle, and 1 G3P, or the only one molecule which has 3 carbons and a phosphate group with it. So only 1 of them leaves. But what happens to the 5 of them? So these 5 molecules eventually go back. They eventually go back to the ribulose biphosphate molecule right here. So they eventually end up being RuBP. So hold on a second. Sorry about that, my brother's phone was ringing. I had to give him his phone. Anyways, sorry for the interruption. We start off with the G3P molecule. Out of 6 of these molecules, only 1 leave the cycle, and the 5 of these return back. So what happens? So these 5 3 carbon chain molecules eventually go back, and they are restructured into, again, a ribulose biphosphate molecule. And how do they happen? And how does it happen? 1 2 3 4 5 molecule, because and you end up with essentially end up with 3 of these molecules. So you can if you count the number of carbons, so you have 3 and 5 carbons right here. So you want 5 times 3 is 15, and here you had 5 molecules of 3 carbon. 5 times 3 is 15, so eventually it's the same thing. And so another 3 molecules of ATP is used in order for this process. So and you can imagine that you might be thinking, okay, one G3P molecule can't. So what's this function? So first of all, why do we make this? Why do we get this molecule at the end? So because I mentioned in the previous video that when the glycolysis video, the glucose is broken down into a G3P molecule. So now a G3P molecule is being used to make glucose. And if when you have three RUBPs, you eventually end up with one of these molecules. If you double it, if you just make it 6, make it here 6, and here would be 12, so and these would be 12 too. So if you just double everything, you essentially end up with 12 of G3P, and you essentially 2 of these molecules are released. So you can imagine, okay, 2 of these molecules are used. 2 G3P can make a glucose molecule. So this cycle, if it happens 2 times, so you are able to generate 2 G3P molecules. And this is pretty much it for the photosynthesis part. We went through all the way from the cellular respiration to the photosynthesis, and hope you found that video useful, and see you in the next video.